young ladies, a special pleasure to have you here.
Okay, so let's go. It's a pleasure to be here, and I thank you to come here and watch me talking about this today. Today I'm going to talk about the work of the data I collected last year when I was here during summertime. As Jim told, I'm a student from University of São Paulo, but I'm a visiting student here. So the first thing everybody always asks me when I tell that I work with chipmunks is, are there chipmunks in Brazil? And then when I say no, there are not, the next thing is always, so why are you studying chipmunks? And I'm studying chipmunks for a lot of reasons. The main reason is because they are really cute. As you can see here. The second reason would be because I knew that I would be able to go and do field work in such beautiful places. Oh, no. This is just to show that it's the spaces that I work in during my master's. So I have improved a lot at this. This is awesome.
They smell bad too.
So, and I knew that I would be able to do field work in such beautiful places like this one. This place here is at Circulate in the Southern Sierra Transect. And we went there this year, me, Carol, and Les and Peggy, and Jim, of course. And the next reason why I would so bad to work here at MVZ is because I knew that I would be able to work with the master of the masters. And, well, everybody who knows him knows that he knows more about mammals than 99.99% of the world. So, it's a pleasure to work with him. And last and not less important, I wanted to be part of the Grinnell Research Project because I think it's a fantastic project. And before I start talking about the Grinnell Research Project, I want to talk a little bit about Grinnell. As everybody here knows, Grinnell was the first director of the MVZ, and he was one of the greatest naturalists of the last century. He was really careful in collecting his data, so we have a very, very good record of all the specimens he collected. We know exactly where it was collected. And so then for California, we have a very broad record of the beginning of the last century made by Grinnell. These dots here are the collection points of Grinnell and his colleagues and students. And this phrase here, and I think that it's everybody here has already heard at least 1,000 times in their life, but it's really, really great. And he wrote in his book in 1910 that the greatest purpose of our museum will not be realized into the lapse of many years, possibly a century. And this is that the children of the future will have access to the original record of fauna conditions in California and the West, wherever we now work. And then, so the MVZ people read that phrase, and it was Craig actually, and he decided to return to the same places where Grinnell has collected and collect again to see how the fauna has changed in California from the last century. So the main goals of the Grinnell Research Project would be to revisit the same places that Grinnell and his colleagues had collected and document changes in communities and relative abundances of species, and then observe the changes that had happened in a century in the genetic diversity, phenotypic diversity, and so on. Understand why the biodiversity has changed given the changes in the environment, and provide a predictive model of how the vertebrates diversity will respond to the continual change. So here just to show how California has changed in the last century. As we can see, the temperature in California has increased a lot in most of the places. In some places has increased as much as 2 degrees Celsius, the median temperature. And coupled with that, the average precipitation has changed in 2. In some places, California has got wetter, but in some places, especially in the eastern slope of the Sierra Nevada, it has got drier. And together with the climate and change, extensive changes have occurred to the landscape. Here are photos from the Grinnell. It's written in Portuguese, I'm sorry. As you can see, this is from Mount Dana. In this slope here, at the beginning of the last century, almost no trees was apparent, and nowadays there are a lot of trees, so the landscape had changed too. And another photo from the Lake Vogelsang, and the same thing, where last century there was almost no trees, nowadays we have a lot of trees. So the question about my presentation would be how two species of chipmunks were affected by these changes, the landscape and climate and environment. The species I'm going to talk about today are two species of chipmunks. My favorite one, Themis alpinus, and the other species is Themis speciosus. Both species occurs in the Sierra Nevada, with Themis alpinus occurring, having a much narrower distribution, occurring only from the central to the southern part of the Sierra Nevada, and Themis speciosus has a much broader distribution occurring throughout the Sierra Nevada. This figure here I took from Grinnell's book, Animal Life in the Yosemite. That's why it's not a very clear picture, but I want to show that. Showing that even at Grinnell times he had a very careful understanding that both the chipmunks species in the Sierra Nevada used to occurs in an altitude gradient. So while alpinus occurs in very high elevations, in the Hudsonian and Alpine life zone, as Grinnell has named it, Themis speciosus and the subspecies that occurs in the Sierra Nevada stratum, it occurs at lower elevations in the Canadian Hudsonian life zone. So I'm going to talk today about two transects that the Grinnell people had worked with. The first transect is the Yosemite transect that has been originally served by Grinnell in 1914 to 20 and reserved from 2003 to 2006, and the Southern Sierra transect that was originally reserved from 1911 to 16 and reserved from 2008 to 10. So, what they found about how the distribution of these two species has changed for Themis alpinus in the Southern Sierra transect, they have had an upward retreat in their distribution, while in the past they used to occur in elevations as low as 8,000 feet. Nowadays they only occur at elevations higher than 10,000 feet. And in the Yosemite transect the same thing has happened, but the upward retreat was a little bit smaller. And on the other hand, for Themis speciosus, in the Yosemite transect, any change happened in their distribution, the altitudinal distribution, and in the Southern Sierra, a slight change upward has happened to Themis speciosus. So Emily Rugby studied during her PhD that they did here the genetic diversity of these two species from the historical and modern era, only for the Yosemite transect, and she found that while in Themis speciosus it has not changed its genetic diversity, she used 8 microsatellite lots. Themis alpinus has changed, it has decreased, has lost genetic diversity. And also Themis alpinus has got more, the populations in Themis alpinus has got more divergent, they got more differentiated. And in Themis speciosus, the FST, that is an index of population differentiation, has not changed. So we have two species of chipmunks with different responses to the habitat and climate change that happened in this last century, while we have Themis alpinus that have shrunk its distribution, the population has become more genetically differentiated and it had a loss of genetic diversity. Themis speciosus has not changed. So the question I was trying to answer with my work was, what is the impact of these changes into the phenotype of these two species? more specifically, I had 4 questions that I did to myself and tried to answer. The first one was, did the morphology change? And I'm gonna answer to you yes. The second was it only in size or if their shape has changed also? The third, did the patterns and magnitude of morphological integration change? And the last one, what evolutionary process was underlying these changes? So the sample that I used come from these both transects. I used from the Yosemite transect 8, 9 Themis alpinus, 51 historical and 38 modern. Themis speciosus, the subspecies that occur in the Yosemite strata. I used 9, 3 historical and 226 modern. And the Southern Sierra transect 6, 7 historical alpinus and 29 modern. And Themis speciosus sequoiensis I use. Sequoiensis is the subspecies that occurs in the Southern Sierra. I use 80 historical and 98 modern. To measure the specimens, I use a digitizer, a microscribe that has like this pen, and I put the pen into the landmark I want to measure and press a button, and then the microscribe sends to the computer the 3D coordinates of this place. I use 48 landmarks in the skull, on both sides of the skull, and for the data I'm gonna analyze using geometric morphometrics. I'm gonna use the landmarks as the traits. Otherwise I'm gonna use 35 linear distance that I calculate from these landmarks as the traits. And here are the 35 landmarks that I use. In red we have the face traits and in blue the neurocranial traits. So going to the first question, did the morphology change? To calculate that, I calculated delta z, that is a vector of morphological change. It's very simple to do that. You just calculate the mean of the modern samples of each trait and subtract the mean of the historical samples. So what I found was that here in this picture I'm gonna plot in red everything that has increased and has contributed to the change in the delta z more than 0.1. So the traits that have changed more, and in blue the traits that had contributed more than 0.1 but has decreased instead of increasing. Here we cannot see any blue lines because everything for Themis alpinus in the Yosemite has increased. So it has increased a lot, and then I thought, okay, and if I use the first principal component, that is an estimation of size in this species, and calculate an ANOVA to see if this change is significant or not. As you can see here, the increase in the size of this species is highly significant with a P of less than 0.001. So they have got more big. And then looking to this picture, I saw that most of the traits that had increased seems to be in the face. And then I thought, okay, and if I separate the traits from the face from the neurocranial and redo an ANOVA to see if the changes were just in the face or is in the neurocranial as well. Don't laugh to me, no. And what I found is that the face has changed a lot. It has got bigger, and the neurocranial has not changed at all for Themis alpinus from the Southern Sierra. From the Yosemite, sorry. And from the Southern Sierra, it's exactly the opposite. It has decreased in most traits, but some traits has increased it also. And this decrease is significant with a P of less than 0.05. And when I separate the face from the neurocranium, the face decrease was significant and the neurocranium was not changing. So it is also changing in the face, but it is decreasing. For Themis speciosus from Yosemite, although they have some change, most of the traits were increasing. It's not significant the size difference. But when I separate the face from the neurocranium, the face is significant with a P of exactly 0.05, and the neurocranium was not changed. And for Thymius speciosus from the southern Sierra, some traits were increasing, some traits were decreasing, but the ANOVA was not significant, not even for the skull as a whole or for the face or the neurocranium. So putting all this together, here are the ANOVAs for the entire skull that I showed. As we can see for Thymius speciosus, they have not changed. And when I plot both of them together, we can see that they were different in the past and they continue to be different nowadays. But for Thymius alpinus from the southern Sierra is decreasing, and from the Yosemite are increasing. And when I plot the size of them in the past, they were very divergent, and they seem to be going to the same place in size. So did they change? Yes, at least for Thymius alpinus they have changed a lot. For Thymius speciosus from the Yosemite, they kind of have changed, in the face at least a little bit, and in the southern Sierra no. So going to the second question, was it only in size or not? So I used geometric morphometrics because it removes the effect of size. When I do the percussive superimposition, it removes the effect of scaling. So I'm going to see only difference in shape and not size. And then I calculate two index of distance, the percussive distance and the Mahalanobis distance. The percussive distance is a measure of shape difference, but it does not take into account the covariance, the covariation between the traits. And the Mahalanobis distance does take into account the covariation between the traits, and it also is a nice index to use because I can compare to other studies, because these studies that use evolutionary chains, they usually calculate the difference into shape or into morphology using units of standard deviation, so I can compare that with my study. So what I found is that for the percussive distance, both Thymius alpinus has changed much more than Thymius speciosus, and the same thing for Mahalanobis distance. But we can see here in the Mahalanobis distance that the shape change in Thymius alpinus from the Yosemite was much higher than for Thymius from the southern Sierra. So no, it was not only in size that they changed, but the shape has changed also, especially in Thymius alpinus. And here I'm plotting the shape change that I observed. Here is just the skull with the landmarks I used, and it's not very good actually. But in gray are the historical shape, and in red are the modern shape. You cannot see the red, right?
Yeah, you can see a little.
Yeah. So as you can see for Thymius alpinus in the Yosemite, the skull seems to get a little bit slender, and there is a lot of shape difference in the face. And this is the one that has changed more in the shape. For Thymius alpinus from the southern Sierra, the difference in the shapes are much more subtle. It seems to have some difference, especially here in the neurocranium. It's getting a little bit small. And for Thymius speciosus, you can barely see any differences. The gray line is almost coincident with the red line, so the shape differences that happen here are very, very subtle. And the same thing for Thymius speciosus from the southern Sierra, with the exception of this place here that seems to be changing a little bit more the face of sequence. And is it this shape difference comparable to other species? As I told you, the usual measure of evolutionary chain is counted as unit of standard deviations per generation. And for example, we have this study here that I'm sure that everybody here has already heard about it. It was done by the Grant couple, with Darwin finches, and they found that during 1977, because of very drought conditions in the island of that these finches used to live, almost 75% of the population has died because of that, and the only part of the population that has survived was the birds that have larger beaks. So in only one generation, the beak has changed in this species about 0.65 standard deviations, and that's a lot. So let's compare with what I found for my chipmunks. So I took the Mahalanobis distance that I have calculated and divide by the number of generations that have passed from the historical time to the modern time, and it's about 9 generations. And what I found is that Vertebrate alpinus from the Yosemite is 0.15, and it's the higher one, and it's much lower than what happened to the Darwin finches. But what happened to the Darwin finches was really, really drastic, as you can see. And so what a population can do when it's facing natural selection, because natural selection imposes a demographic cost in this population, they have 2 options. Or they can get adapt, change and get adapt, or then if the cost is too high, they go extinct. And Berger and Lynch in 1995, they did theoretical models using, it's theoretical. And they predict that into a changing environment, the maximum sustainable rate of phenotypic change would be 0.1 standard deviation per generation. Anything that was higher than that in their models, that has a lot of assumptions, of course, everything that was higher than 0.1 went extinct. So coming back to Metemus, we can see that at least for Vertebrate alpinus in the Yosemite, it's higher than the theoretical maximum sustainable rate. So it means that they are changing a lot, and it's really drastic. And it can be much more drastic if we think that the environmental change that probably is causing all these morphology changes has not started exactly 90 generations ago. It's probably has started much more recent. So probably Vertebrate alpinus from the southern Sierra is changed at a higher rate than the maximum theoretical environmental rate. I'm not saying that this population is going to be extinct. Who is saying that is linked? No, I'm kidding. But it's very, very drastic. So going to the third question, did the patterns and magnitude of morphological integration change? Well, what's morphological integration? Morphological integration refers to the relationship and connections among morphological elements. So because they have an underlying genetic architecture, they share the same function, or they share the same developmental origin, morphological traits tend to co-vary and evolve together. And that goes to the notion of modularity, that morphological traits that co-vary forms a module, so they tend to evolve together. But there are some traits that are in other modules and does not have a covariation with the other traits. I'm going to try to explain this better. I just want to call attention that this morphological integration is essential to understand the evolution of complex morphology. So looking to the Sayuri skull, if you take 2 measurements, the first measurement of the maxillary length and the second measurement of the mandibular length, if we plot these 2 measurements together, we're going to see that they are highly correlated between each other. And why would that be? Well, there's a lot of hypotheses of why morphological traits are correlated. The first one would be because they share the same function, and for these traits we know that it's true. Both are related to feeding. They share the same developmental origin, and we also know that it is true for these two traits. They came from the neurocrests, and or they share same genetic architecture. Genes that affect one trait might be affecting the other one. Pleiotropic effects. And on the other hand, we expect that traits that do not share the same function or developmental origins would have lower correlations between them. So if you take the maxillary length again and measure the parietal bone and plot these two measurements together, we expect that they have a lower or absent correlation between these two traits. And so we say that they are in different modules because they have different functions. While the parietal is involved with protecting the brain, the maxillary is involved in feeding. And they share, they have different developmental origins. The parietal comes from the paraxial mesoderm, and the maxillary comes from neurocrest. So why should we care about it? Mainly because the more phenotypic integrated an organism is, the more constrained to respond in the direction of selection. So if you have selection pushing to the maxillary to get bigger, if the skull is too constrained, if the correlations are too tight, you cannot just change the maxillary. You have to change all the skull in order to change that. So the degree in which the traits are correlated is really important for evolution. So how to calculate the magnitude of morphological integration? There are several ways of doing that. I use an R-squared, that's an index of overall phenotypic integration. The higher the correlation between the traits, the more correlated, the bigger is going to be the R-squared. So what would I expect to happen to the morphological integration, to the R-squared, between the modern and historical samples of Thamias? Well, first of all, I would expect that nothing happened. The same morphological integration index they had in the past is going to be now. But it's the null hypothesis for a population that had remained stable with any change in its genetic architecture, morphology, population size, and so on. So that's my hypothesis for what's going on in Thamias speciosus. They are not going to change their morphological integration. On the other hand, I would expect a decrease in the morphological integration index for populations under stressful conditions. This study here is the only study that I found studying morphological integration into populations and not species or genus or so on. And they studied 4 species of shrews. I'm only plotting here. And they showed that for shrews that have grown in control habitat, in stable habitat, the morphological integration index was higher than shrews that have grown into extreme environment, deforested areas. So as we're seeing that Thamias alpinus is changing a lot and probably is under stressful conditions, I would expect that Thamias alpinus would decrease their morphological integration. And what I found was exactly the opposite. For Thamias alpinus in the Yosemite, this is the 95% confidence interval calculated with a bootstrap. I found that the red are the historical, and the modern are blue. And I found that the morphological integration has changed significantly for this population of Thamias alpinus. So instead of getting less morphological integrated, they have gotten more. But my sample size for the modern sample is only 38 individuals, which might lead to an upward bias in this estimation. So I would need more samples for, and I'm looking to you for a reason. I would need more samples from Yosemite to see if this pattern is real or not. And for Thamias alpinus from the Southern Sierra, although there is a trend of increasing in morphological integration index also, it's not significant, so I conclude that they have not changed their magnitudes of morphological integration. But once again, my sample for the Southern Sierra of Themis alpinus is only of 29 individuals. And for Themis speciosus on both transects, they have not changed significant their morphological integration index, and that was as I expected in my hypothesis. So, putting everything together, Themis speciosus and alpinus from the Southern Sierra has not changed their magnitude of morphological integration, and that's what I would expect in a stable population. And Themis alpinus from the Yosemite has increased instead of decreased their levels of morphological integration. And it has already been shown that that directional natural selection can increase morphological integration. So I think that's an indication of natural selection in this population. And what about the patterns of molarity? So, and how are there modules in this species, or they skull comparts as a whole? So what models I would expect in Themis, because we saw that the face seems to be changing decoupled from the neurocranium, I would expect at least two, the neurocranium and the face. And then I asked it if the patterns of modularity have changed in the last century or not. How can I do that? Well, imagine here is an illustration with only 3 traits in each module. Imagine that I measure these 3 traits here in the face. I expected that if the face is a module, the correlations between these traits would be higher than the correlation between the traits in the face and in the neurocranium. And the same thing for the neurocranium. I expect that the correlation would be higher in the neurocranium if it is a module. And I can test this doing a hypothetical matrix, where I put the number 1 into the traits that I expect to be in the same module, and number of 0 in the traits that I expect to be between modules, and correlate this hypothetical matrix with my real matrix, doing a mental test to calculate the p-value. And if it's significant, it means that they share the same structure, meaning that they have modules. So I have 2 hypotheses, 2 hypothetical matrices, one that I have the neurocranium as a module, and one that I have the face as a module. And what I found for Themis speciosus, once again, is that they have not changed a lot. That's why they're not my favorite. And they don't have any modules, not even in the face or in the neurocranium. But for Themis alpinus, it's really interesting that in the Southern Sierra, the face is a module in the historical time and in the modern historical, and the neurocranium not. And more interesting is that Themis alpinus from the Yosemite, the face was not a module in the past and has become a module nowadays. And it's really, really cool because some simulations made by a friend of mine at my lab in São Paulo, Jogo Melo, has showed that this pattern of arising of a module is exactly what you would expect if selection would be acting in these face traits. So summing up, Themis alpinus from the Yosemite, they have changed not only the pattern but the magnitude of morphological integration, suggesting that something is going on, probably natural selection. Themis alpinus from the Southern Sierra have not changed, not even their magnitude or pattern, but they do have a module already in the face. And Themis speciosus has not changed again anything. So going to the last questions, what evolutionary process was responsible for these changes? I use a univariate drift test, meaning that I'm going to test selection for each trait separately, proposed by Lynch in 1990. And that's very simple. It's just calculate a delta that is calculated using the variance between groups, divided by the variance within groups times the time of separation of the 2 species. And compare this delta with the amount of divergent expect under neutral evolution, that would be K. So K for morphological traits under genetic drift is expected to be between 10⁻⁴ to 10⁻². Anything that lies within these values, I assume that genetic drift was responsible for that. Something that is changing more than 10⁻² would be an indication that directional selection is responsible for that, and anything changing less than 10⁻⁴ would be an indication of stabilizing selection. And what I found is it's really cool. Vertebrate alpinus from the Yosemite and the Southern Sierra. I'm gonna plot in this plot. Red is everything that has changed more than expected by drift, all the traits that have changed more, meaning all the traits that have rejected the null hypothesis of neutral evolution. So most of the traits had rejected the hypothesis of neutral evolution changing more than expected by drift. And in blue are the traits that have changed less than expected by drift, indicating that stabilizing selection was acting on that. So as you can see for Vertebrate alpinus, most have changed more. Most of the traits that had reject drift were in the face, once again. And most of the traits that had not reject drift were in the neurocranium. And for Vertebrate speciosus, only looking to the picture, we can see that there are much less traits that had rejected the null hypothesis of genetic drift. So most traits were evolving according to drift, and most that had reject drift were in the face, even though for Vertebrate speciosus from the Yosemite, and for Vertebrate speciosus from the Southern Sierra, very, very few traits had rejected the null hypothesis. So we have for Vertebrate speciosus that most traits had changed in an amount that would be expected under genetic drift, so I assume this species is evolving according to drift. But for Vertebrate alpinus, most traits have changed much more than expected by neutral evolution. And that's an indication of natural selection, directional selection. But then I thought that maybe phenotypic plasticity could be the pattern responsible for all these changes I was seeing. So how can I disentangle this two process? And I do not have an answer to that. But although it's really clear to me that natural selection could be responsible for all the changes I observed, for the changes in shape, in size, for the changes in morphological integration index, for the changes in modules, and not even to say the drift test. It's less clear if phenotypic plasticity could do that also. So the question I was doing to myself is if phenotypic plasticity could be responsible for all the changes I observed, and I don't know. But I thought that maybe a way to test this would be like, as I showed at the very beginning, that Vertebrate alpinus were very divergent in size and that have become more similar. So if the only way that phenotypic plasticity could account for this change, they were divergent and become more similar, would be that that in the past the environment was more dissimilar, was more divergent in both transcends, and had become more similar. And I think that I can test this with the data they have in the Grinnell Observatory. And I would be really happy if you guys have another suggestion of how I can test this. So just to conclude, Vertebrate speciosus has not changed their morphology at all, and not even its distribution and genetic diversity, so I conclude that it's a stable population. And Vertebrate alpinus has changed everything, its size, face, shape, morphological integration, at least for Yosemite, and it seems to be evolving according to natural selection. And it also has changed its distribution and genetic diversity, meaning that's a changing population. So there is a relationship between the observed patterns and distribution of change and morphological change in Thames alpinus. So it's a more sensitive species to the environmental change that occurred in the last century. So although I have named my talk as effects of climate change, I'm not sure if it was climate change that was causing that. And that's the really cool thing about the Greenhouse Reserve project, because it has people who is studying the ecology of these chipmunks. Rachel is studying. And I think with her data we can separate the amount of landscape change and climate change that had driven these changes in. So the next things that I want to do with this data is first develop a multivariate test of genetic drift, something that tests the skull as a whole and not one trait at a time. Disentangle phenotypic plasticity from directional selection. Analyze this data using geometric morphometrics with the help of median cells. She helped me a lot already. I spent 1 week with her in Michigan. She taught me a lot of stuff, but I wasn't able to put everything in this talk because I didn't have time. And last, I expand the number of species and specimens I studied. So this is the people I want to thank.
Somebody can hit the lights. I'm sure Anna Paula would be delighted to answer any questions.
I have a question about your calculation of number of generations between the historical and modern. It sounded like you said there were 9 generations, is that right?
90.
90. Oh, that's a big difference, yeah. 90.
It's the English.
Thank you for that.
I was just going to say, I feel for you with the comment about phenotypic plasticity and not knowing, because you essentially have 2 time data points then and now. I have my lab mate and I and one of our undergrads are looking at some lizards, and we're finding that the differences in, it's a geometric morphometrics project, we're finding that the year-to-year differences are greater than any other difference, even though we're collecting from the same site every year. So I guess this won't be surprising. I suggest killing 30 chipmunks a year. No, I think that it may actually, and on a serious note, I think that it may take that kind of collection, like a focused, targeted collection over a series of years from the same site to get the type of data.
Yeah, I was thinking of doing some common garden experiments with Telly, but for me it's kind of hard to be here every year because I'm from Brazil.
Right.
And
Well, no.
and I need money and I need time and I need.
Right, of course. It's beyond the scope of your dissertation
Yeah.
as it is now. I'm just saying, like.
But yeah, it would be awesome to do something like that.
Can you see any differences in the habitats? And for example, did they record food from stomachs of these animals or those sorts of things?
We have a specialist on that sitting there.
I'm doing the 3 Grinnell resurvey sites, and I have currently 2 years of data from each of those sites, and I'm taking data on not only microhabitat characteristics, like qualitatively, like number of trees and things like that, but also attempting to ID plant species. And I have 1 more field season planned, so I will have that data from a small number of sites.
But has anyone looked at difference in foods? I mean, you're looking at the front end of the skull, and you would think it'd have something to do with gathering foods as selective mechanism. And so I'm just wondering, is anybody tracking that, or is that below the radar screen?
The data that we do have to address that question is hair samples. You can get stable isotope values from them. I've done a little bit of looking through the herbarium records, and I haven't found a good kind of match between where the historic specimens were collected and samples that are available. So it's a little bit difficult to get that from the historic data, but you can take hair samples directly from the chipmunks, and I've done that for the chipmunks from Yosemite, and I'm currently working on chipmunks from the Southern Sierras.
So I'm sorry, I just have to ask, what's the speculation though about the food handling differences, since it's all mostly in the face and potential convergence? Is there
It's kind of hard to answer that because it depends of how the diet has changed. So if we saw that maybe they're having harder food nowadays, I can look if the face, the muscles in the face had reorganized or something like that. But it's kind of hard to do that.
You could theoretically calculate muscle force just from those measurements, changes in muscle force, to know whether or not that change is increasing or decreasing as the face gets bigger. And I don't know that. So it could be they're selecting for softer foods today than in the past, or it could be that they're selecting for harder foods, or they're capable of handling harder foods. But you could theoretically calculate that.
I was just thinking with relation to your sort of climate change landscape aspect, I don't know if any of these have a large enough elevation that you have more than one site.
Sorry.
So if you have an elevational transect where any of your species, like speciosus, has something at 800 meters and 1,000 meters and 1,200 meters, if you then compared modern and historical for each one of those sites, if it is climate, you would expect a trend in one or other direction. So you're pooling everything together now. What I'm saying is for some of your taxa, like speciosus, you may be actually swamping any subtle signal that there might be by putting everything together across your transect. It might be fun to see if you have enough power to break it out as you go up the transect, to see whether the ones at the top are doing something different to the ones at the bottom. It might not be possible with sample size, but it might be something worth looking at.
I've tried to do that, but my sample size, it's not very good to do, like,
Yeah,
very
it's always-
2 years. But I did that. I saw because the first thing that came to me was, okay, they're getting bigger, maybe that's just because only the higher elevational ones were the ones that were survived, but that's not the case. They don't have a
There's no
trend
trend.
into size, at least from the elevational gradient. But that, yeah, okay. So you're saying to me to separate them and just...
Yeah, and you could even go a step further with Emily's data. You could calculate the extent of gene flow between pairwise sites. That would give you a direct measure of selection or probable selection. So you could use that as a direct estimate of new alleles coming in versus morphological change and compare those two. And that'll give you an independent way to look at where the selections might be, whether that's higher than what you would expect, for selection. But that's dependent on whether there are morphological changes stepwise across the gradient.
Legs first.
Oh, do you have any evidence, I'm sorry, in the one that had a lot more change, do you have any evidence of diet change? Did you?
The stable isotope signatures in Alpinus show a decrease in the nitrogen over time, which is likely to indicate, or what Todd Dawson has told me it might indicate is an increasing reliance on nitrogen-fixing plants. But it's kind of hard, I think, to distinguish. I mean, I have a lot more paired historic and modern sites for speciosus than I do for Alpinus. So it's kind of hard to see if that's like an actual change is related to food resource, or it's just random and my sample size happens to be larger for species.
I was thinking more of a what they are eating, not from a isotope standpoint, but for how hard it is to chew standpoint. Because you talked about the parietal being not functionally linked, because you listed it as a brain case bone, but the temporalis muscle, which is a major muscle of chewing, it originates on the parietal. So they're not functionally, I mean, yes, they have separate functions, but they're also united in that function. And if you were trying to chop down, perhaps tooth-wear studies or looking at the morphometrics of the tooth, it might be able to contribute to explaining that. Because if you were eating something that's the physical properties of it were different, and I could see, if it's a radical change in a short amount of time, I could see that possibly expressing.
So remember though that these are rodents, so most of the chewing action comes
It's
from the
within
masseters,
masseter.
not the temporalis.
That's true.
Even though squirrels have a bigger temporalis than most rodents do.
Okay, I think let's stop there. Thank you very much for a very interesting talk.